Hi, everyone. Thank you so much for joining us today. This is the first HBI Harvard Brain Science Initiative 101 talk of the new academic year. So thank you to everyone joining us on Zoom, as well as everyone joining us in person. I'm really excited today to introduce today's speaker, Dr. Christiane Wrann, who is an assistant professor in medicine. She works at the Cardiovascular Research Center at Mass General Hospital and the McCance Center for Brain Health at Mass General Hospital, which you'll be hearing a little more about in a minute, as well as HMS. She leads the program in neuroprotection and exercise. She's an affiliate of the Harvard Stem Cell Institute, as well as a faculty member in the program in neuroscience. Her research focuses on the beneficial effects of exercise and metabolism in the brain. She's particularly interested in secreted factors that are important in exercise and could be potential drug targets. Dr. Wrann studied veterinary medicine at the University of Veterinary Medicine, Hanover, the University of Cambridge, and Cornell University. She has her PhD in immunology from the University of Veterinary Medicine, Hanover. She did postdoctoral work here at Harvard Medical School and Dana-Farber Cancer Institute in the lab of Dr. Bruce Spiegelman. And she started her own lab in 2016. And we're really excited to have her here today. Just a couple of housekeeping announcements. We are aware that there is a graduate student event at 2 p.m. So this talk is going on with questions until 2:15. But if you are a first-year student who needs to run to that event, please feel free. What else? And in terms of questions, if you're joining on Zoom, please feel free to type questions in the chat at any point during the event. We will have an extended Q&A at the end. And I wanted to take a couple minutes to say hi to Akshata Soni, who's the scientific program director for the McCance Center, and she's going to tell us a little bit more about the center. We're looking for ways that the Harvard Brain Science Initiative and McCance can work together.
Thank you, Parizad. And good afternoon, everyone. Thank you for joining us. It's my honor to provide a second introduction to Dr. Christiane Wrann. Nearly 4 years ago, the McCance Center was launched with the mission to reduce human suffering and maximize human potential through better brain health. The three founders that you see up on the screen, neurologist Jonathan Rosand, neuroscientist Rudy Tanzi, and psychiatrist Greg Friccione, shared the belief that the brain health crisis can only be solved through broad, multidisciplinary collaboration and creativity. Arresting the progression of diseases like dementia, depression, or stroke once they've begun is just not enough. We need to prevent these diseases from occurring in the first place, and prevention is possible. Lifestyle choices and proper treatment of conditions like hypertension can dramatically reduce our risk. And by some estimates, if these steps were taken for all of us, the number of people affected by dementia could be cut by 1/3. The McCance Center's Spark funding program seeks to support the most promising research in brain health. That is, research that shakes the status quo from one that is merely proactive to brain disease, one that's merely reactive to brain disease to one that is proactive and preventative. Dr. Christiane Wrann was the recipient of one such Spark Award that was given to support her work in understanding the molecular mechanisms underlying the benefits of exercise as an intervention in dementia. Her findings have been groundbreaking, featured in the New York Times, the National Geographic, the Economic Times, amongst others. Her work has also received recognition from the NIH, and most recently, in August, has led to competitive funding through the Alzheimer's Drug Discovery Foundation. We look forward to seeing how Dr. Wrann's important work impacts the way we treat dementias and preserve brain health and prevent brain disease in our population. So thank you again, and take it away, Dr. Wrann.
Yeah, thank you so much. Yeah, thank you so much for the kind introduction. It's really a great pleasure to be here today to present the science, present our work, but also to be back in an in-person event and kind of catch up with everyone after the last 2 years. So I'm really excited about the opportunity to kind of bring scientists back together. All right, so I just always have to just show briefly my disclosures as mandated by MGH and Harvard University. All right, so dig in. So when we think about the beneficial effects of exercise, what everybody is acutely aware of is, of course, their effects on muscle strength, on muscle mass. Most people also know about the benefits on cardiac health, cardiovascular health, improvement in metabolism, for example, for patients suffering from type 2 diabetes. However, there is more and more research really showing that exercise is also good for your brain. And that's mainly due to two reasons. One that Akshay already has mentioned is that exercise is attacking several of what we call modifiable lifestyle factors or risk factors for disease. So this includes, for example, smoking, obesity, and most importantly, high blood pressure. On the other hand, exercise also has intrinsic effects on the brain. And that's the part that got me really excited in this work when I started out, to kind of understand how something that seems to be so far from the brain, like, for example, like as running exercise, is actually doing things in your brain and leads to this amazing plasticity that we all have. So, and this is what we're going to talk more about now next. All right, so to first clarify something, like, what is exercise? So when you're an exercise physiologist, we distinguish between what is called physical activity. So this is all the activities you do that involves, working muscles in your body that gets you above resting metabolism. So this includes, for example, daily activities such as cleaning, gardening, walking your dog, walking to work. And then we have what is formally called exercise. So this is a structured and planned type of physical activity with the aim to maintain or improve physical fitness. And there again, we have the endurance or also aerobic exercise, which is aimed to improve cardiorespiratory fitness. So, this is like everything that basically gets your heart rate up and gets you out of breath. This includes running, hiking, biking, swimming. My personal favorite is the hiking part. I really love hiking the beautiful New England. And the other thing also is kind of saying in that is that there's also work that shows that exercising outdoors in green spaces has also calming effects on your mood and your stress level. So you might be getting other benefits there as well. And then, of course, we have resistance exercise, which is improving your muscle strength. So this is the classic weightlifting, but people who have done high intensity yoga class can also tell you that there are other exercises that can get your muscle strength up. And interestingly, so the evidence that we have for improving brain health specifically, the evidence we have from mouse studies and from human studies is that really aerobic exercise is having the most benefits. This is not to say that you shouldn't do anything for improving your muscle mass, especially when we think about an aging population. There's a huge risk of falling. So keeping up your muscle strength, your flexibility, your balance is also equally important. So what does exercise do for cognitive function? So the one thing what it really can help with is improving cognitive function in older adults. This is a study done at the McCance Center by colleagues Wrann and Westover, where they was aimed at actually an aging population that has been currently sedentary. And the exciting findings that they had is with an exercise program over 12 weeks where the participants could choose the modality. So you didn't have to do a certain exercise. They could do at home whatever they want using a heart rate monitoring device, as long as they kept up the heart rate at 50 to 75% max heart rate. And the study nicely showed that the participants overall improved their VO2 max. So this is a measure of how much, what's your aerobic exercise capacity, your aerobic fitness level. And that this also led to an improvement in cognitive function in like a mixed cognitive function task. And again, something interesting detail there is the improvement in VO2 was positively correlated with cognitive enhancement. So the more you improve your VO2 max, the more improvements you saw in cognitive function. And I will bring this topic more, but there is a dose-dependent effect for the benefits of exercise. So what about Alzheimer's disease? So there is really good work out there that shows that more physical activity is a good preventatory measure. So on the one hand, we have data, for example, from the Rush Memory and Aging Project, which was a really large study where they outfitted participants with these Fitbit devices and then just monitored over the 5 years how much activity you do and who's developing dementia. And depending if you're in the top 10% or bottom 10% of physical activity, you cut your risk of developing dementia into half. And there's just a new study came out, I think, a week ago in JAMA Neurology, where they show that as little as 4,000 steps a day can already reduce your risk of dementia by 25%, and the maximal effect was close to the 10,000 steps you hear about a lot, which cuts your risk of dementia in half. So what about for patients that already have developed Alzheimer's disease? This is data from what we call a meta-analysis of different studies, and you can see the red line is basically neutral, and then depending on if it goes further to the right, you see beneficial effects on exercise. If it goes to the other side, you see a reduction in cognitive function with exercise. And you can see that the vast majority of studies had strong effects on improving cognitive function. The data is a little bit more mixed in dementia, because anyone who is a clinician neurologist can tell you the people who are getting dementia, it's a very heterogeneous population, and it really does matter like where in the disease you're catching people. And the another thing is also, there are very different exercise regimens. So again, the leading theme was that exercise intervention that did not have an aerobic component did not lead to improvements in cognitive functions. And again, improvements in CFR, this is what we call the cardiorespiratory fitness, the readout, how much you pushed yourself in exercise, was positive related with cognitive enhancement. What else is exercise doing to your brain? So on a kind of like a structural level, one of the first really exciting findings was that exercise actually can help you to preserve brain tissue, brain volume. So this was shown in this, again, aging population where they had a strenuous walking exercise several times a week. I believe it was 5 times a week. And then over the course of the year, they came back with MRI to measure hippocampal volume. And you can see that the participants who didn't exercise actually lost 2% of the hippocampal volume. And the participants who did gained 2% in the hippocampal volume. And the other reason why I show this is that a lot of the exercise effects we are seeing are actually happening in the hippocampus, which you probably all know, are very well aware of, is an important center for learning and memory, specifically spatial navigation. It's one of the centers in the brain that gets very early affected in Alzheimer and aging. So that's why, again, it's this exciting finding that exercise is strengthening those areas of the brain that are most affected in these conditions. And in line with that, also, again, there was an improvement in the spatial memory task in these participants. What else is exercise doing? So we talked about the increase in hippocampal volume. One other thing it's doing, it's increasing what we call cerebral blood flow. So the amount of blood that actually is going into the brain, and specifically again into the hippocampus. So this is data again from an MRI study where you can look at blood flow. And what they showed again is this nice correlation between how much exercise and how much benefits you get. So the first graph is what they call the VO2 max. So again, how much did you improve your aerobic fitness level? And then they look at the change of blood flow to the hippocampus, and you see this nice significant positive correlation. The same then holds true for changes in hippocampal volume. And then lastly, cognitive function tests showed the correlation as well, which suggests again that one of the mechanisms is actually increased blood flow, which makes sense because hypoperfusion is some of the early signs in aging and in Alzheimer's disease that is happening. And hypoperfusion, of course, leads to a lack of oxygen, a lack of nutrients, and buildup of metabolic products. And this is data now from a mouse study where they showed that it's not just that you have more blood flow to the hippocampus, you also see an increase in angiogenesis. Here they looked actually at the perimeter of blood vessels, but you also see that there's more formation of new blood vessels with exercise. And again, dose-dependent manner. Okay, so since I introduced the mice, so this is nice that we can study this in human. Can we this actually model in mice? And the good answer for someone who's a basic scientist like myself, yes, we can actually model exercise effects very well in mice. So it's one of the few things where mice are actually good model for. And this was first shown by Henriette van Praag when she was working in Rusty Gage's lab. And what they did is this is a typical exercise setup. You place mice into a cage with running wheels. The mice will exercise several kilometers a night voluntarily, 5 to 7 kilometers. It's quite amazing. What we don't know is why placing a treadmill into the office of a human being not has the same effect, but that's a topic for a different day. So, and then you look at these mice, so you have the control mice or the sedentary control group, you have the runners, and then if you do the Morse-Water maze, which is one of the most established cognitive function tests for spatial learning and memory, you can see a good improvement in the learning. So what they have to do in this test is you have this water tank, the mice are swimming in there and they have to find this hidden platform. And then you can measure how long does it take them over, training session of 6 days to learn to find this hidden platform. And you can see what we call the latency. It's significantly faster in the runner mice. And then if you do a probe trial to test for memory, so we take out the platform, let the mice look for the platform. And if the mice has a good memory, it will remember, oh, this was the quadrant where the platform was. I'm absolutely sure about this. So they're just swimming, circling it, hoping to find it. And again, you can see the runner mice had a significant better memory of the location of the hidden platform than the control mice. So what about aged mice? And someone actually had asked me, like, so what about aged if you have never exercised before? These were these mice. So these mice had never exercised before in their life. They were around 20 months old, which, as you probably know, for a mouse is really old. They again did the running wheel exercise intervention. This is again the water maze, and you can see how the non-sedentary controls have really difficulties finding the path even after several days of training, and the runner mice, they have learned it very well. So, which is good news for aged mice, but also good news for aged human individuals thinking about taking up exercise interventions. Then what about Alzheimer's disease? So this is kind of the disease where we are really interested in. Again, we do see these improvements with exercise and cognitive function, mainly again also in the hippocampal dependent tasks such as the Morse-Water maze. I just picked here 2 different examples to make the point that people who work in the Alzheimer field are probably aware of it. These different Alzheimer disease mouse models, these are genetic mouse models, they all have their different shortcomings and advantages. So I wanted to kind of show that this phenomenon is generalizable. So this is one of the study where they use the same, the Morse-Water maze. This is a really nice study from my colleagues Si-Hoon Choi and Rudy Tanzi over at MGH, where they use the radial arm maze and a very severe mouse model of Alzheimer's disease, where they also show this important effect of exercise on improvement in cognitive function. What about AD pathology? To just kind of remind people what are the hallmarks of Alzheimer's disease pathology. So on the one hand, we have these amyloid A-beta plaques that is these like insoluble protein aggregates made out of these APP precursor protein downstream that is, like some people say it's like glue in the brain or something, like these sticky plaques that you accumulate. Then you have the tau tangles composed of hyperphosphorylated tau, and there's more and more research kind of trying to understand, how these like different pathologies interact and make each other worse in the game. And then there's more and more research also now showing that something that these trigger is what's called neuroinflammation. So a response of immune cells in the brain trying to get rid of this pathology, and that this might be actually what kills the neurons in the end, the friendly fire, the trying to get rid of a plaque or this aggregated tau, and thereby killing synaptic connections in the end, the neurons itself. And I'm going to come back to this later, a little bit on the neuroinflammation, so you might going to see the slide again. All right, so in mouse models, again, it's much easier to look at these than in humans. And there it's, again, across vast different mouse models, we see a nice reduction in these amyloid plaques. So these are these brown participates that you see here. Again, this is the dentate gyrus of the hippocampus. So, and C is the control mice, E is the exercise mice. So nice effects. For tau, there's a little bit less data also because there are not as many good Alzheimer or tau models, but across Alzheimer mouse models, they do develop some kind of tau aspects and a lot of tauopathy models. You can see that exercise here, control versus E, there's a reduction in phosphorylated tau. And I just mentioned the tau tangles are being made up of hyperphosphorylated tau. And also there's more and more work showing that neuroinflammation is being monitored. And there was a nice study in Nature earlier, I think it's actually 2021, I believe, not 22, However, I believe actually that the effects, especially on neuroinflammation, are not well enough understood. So this is also one focus in our lab to really understand what are the different immune cells are doing in Alzheimer and how actually exercise is helped to reshape them. So, hope to give you an update sometime soon, or if you're interested in learning more about that, our lab is working on that. So there's another really interesting mechanism of neuroplasticity in the brain, which sparked a lot of imagination, and that's the enhancement of adult hippocampal neurogenesis. So you might remember from your neurobiology classes, in the adult brain, there's not much neurogenesis going on. Exactly, there are only two places where it takes place in an adult mouse and human, and one of them is the subgranular zone in the dentate gyrus of the hippocampus. So there we have stem cells, and the exciting part is that running exercise is one of the strongest stimulators to get these cells to proliferate, then also to survive and to form new neurons and get integrated into the circuits. Again, groundbreaking work by Henriette van Praag. All these black dots that you see are BRDU labeled cells. BRDU labels, when the cell is dividing, gets incorporated into the DNA. You can see there's usually we see effects about 5-fold increases in proliferating cells. If you co-stain them for markers of neurons, such as Nguyen, you can show that these are actually neurons. It's not just glial cells that are proliferating. These cells are actually neurons. And using, for example, a reporter virus that gets integrated into these stem cells, we see that these neurons are actually, they actually make neurons that get fully integrated into the circuit. And there's some work later I'm going to show from our own lab also that even exercise, not just that you get more neurons, but it also changes the plasticity of these neurons. And there's some data that, for example, if you measure long-term potentiation, like a measure for synaptic plasticity in the dentate gyrus, you actually see an enhancement with exercise due to these neuron changes in the brain. This was, I forgot, this was 1999. Now we have a lot more work to really understand how neurogenesis in the adult brain is happening. We have these different stages, which are the transcription factors that are mediating this. We have a better understanding of the regulation. And the 2 points I really would like to make is it's not just that you have more neurons in the brain, and that's maybe great. It's also that these neurons, newborn neurons, have crucial function in the brain in cognitive processes. So the process they're mainly involved is what we call pattern separation. So this is the task, we call maybe spot the difference task, or where did I park my parked my car yesterday and today. So you have these two competing memories, and you're trying to make from these two very similar memories, you try to create distinct memories. And this is where adult-born neurons can help you. And there's really good data to show that all the interventions that reduce adult hippocampal neurogenesis could be with genetic models, could be irradiation, could be stress, could be high-fat diet, Alzheimer's disease, leads to a reduction in cognitive function, specifically in pattern separation tasks. And on the other hand, if you do things that are good to enhance adult hippocampal neurogenesis, such as running exercise, genetic models, pharmacological models, to increase the number of neurons or the neurons that you have to make them, function better, you get improvement in these cognitive function tasks. And pattern separation is also one of these cognitive tasks listed very early on in aging and Alzheimer's disease. So this is one of these things, this like, where did I put my key? So this is why we also believe, for this functional correlate, that there's an importance in the pathology in those diseases. And jumping right back there to like, what about humans? I showed you all this data in mice. Obviously we can take serial biopsies, brain biopsies in humans. And so there's a large body of data that has looked at, neuronal stem cells in humans. This was like one of the first works where they really used, in this case, histological methods to look at different stages of Alzheimer's disease. So these Braak stages are different severity of Alzheimer. So, and you can see the more severe it becomes, the fewer immature neurons you have. And this paper was published, I think, a month ago or something. It came officially out, where they used single cell sequencing of the hippocampus to kind of define a molecular signature of these immature neurons, to look at that in humans and then also in Alzheimer's disease. And you can see that there was a significant reduction of what they call the immature neurons in control versus Alzheimer's disease patients. And also you can see that possibly on a functional level, the neurons might be different as well. So there's good data that this is also not just interesting plasticity we can study in mice, but it actually has pathophysiological consequences in humans. However, we don't have a good assay or biomarker to really monitor adult hippocampal neurogenesis in humans over time. And this is something where a lot of labs are working on, including ours, kind of come up with something that, greatest thing would be, you get some blood drawn and they can tell you, how good are your, is your neurogenesis still? And if you do an intervention, is it actually getting better? So again, new interesting research, you could get involved with. Okay, so I have talked a lot about hippocampal plasticity. So how can we actually start translating something from mice to humans? I already had pointed out that some of the limitations we have in humans is the availability to do manipulations and then also to collect tissue. And in mice, we have the luxury that we can do different interventions and at different time points, collect the tissue cells of our choice. Obviously, cerebral biopsies in humans are neither ethical nor I mean, that's just not something that's going to happen. So most of the technology we can use in humans is non-invasive technology, such as imaging technology. And on the other hand, in mice, it's because it's much easier to do these interventions. A lot of times you have two different groups, but you don't do what we call a longitudinal studies, what is usually what we do in humans, where we start out with humans and then we do an intervention and then we compare before and after. That's what we call a longitudinal design. So this was a challenge that I was thinking about and teamed up with a colleague, Wrann, who at that time was still at MGH in the CVSC and the Martino Center, who is an MRI technologist. So he had developed a really cool imaging algorithm based on what is called diffusion tensor imaging. So this is he works on heart. So this is a picture of a heart, and these different colorations is basically the microstructure of the heart that you can make visible using MRI technologies. And this diffusion tensor imaging, if someone has deep questions, happy to answer this later. But basically, what you look at is how ordered or unordered are the water atoms in the tissue. And then you can basically look at how the spin is changing, like in the the vectors in space. You can look at them. And so this is one example. When you look at gray matter, there is usually less order, so the three vectors have different relation to another than if you, for example, look at white matter, where you have more structure and you have less what we call anisotropy. So this is one technology where we can now non-invasively monitor changes in the brain. So this is mainly used in humans. So we wanted to know, as a proof of principle, can we use this in mice? So we actually have the same technology in mice and humans and start translating some of our research. So we did this in young, healthy, wild-type mice. They were exercised for 0, 30, and 60 days, and we did MRI imaging at these time points. And what we found is, on the one hand, we saw this really, nice increase in hippocampal volume, which I already had showed you was well documented in humans. So this is just for people who are not as familiar with MRI images. This is what is called T2 weighted. So, this is a coronal section of the hippocampus of the mouse brain. You can see the hippocampus here. You can already guess the resolution in a mouse brain, because it's so small, is not good enough to look at hippocampal subfields. That's easier. In humans, the brain is much bigger. Here, basically, one pixel is already like half the dentate gyrus. And then we have the DTI imaging, the color coding laid over that in the hippocampus. And you can see that also we saw an increase in fractional anisotropy. So this indicates that we have a more ordered microstructure in the hippocampal formation with exercise. This could be because there are more fiber bundles. It could be that there's a reordering of existing microstructure, for example, more synaptic connections being formed. And we just recently also published a study to show that kind of similar holds true for also cardiac microstructure with exercise. All right. Something else that caught my attention when I was in Bruce Spiegelman's lab during my postdoc is, again, going back to the question, why is it running exercise that's so good for your brain? Since it's quite far away. And there's these, there were 2 studies published where they actually compared running wheels and enriched environment. You probably have heard about enriched environments in mice. So this is where they have these cages that are outfitted with a lot of toys, places to hide, something to climb on. They usually also include a running wheel. And it was pretty clear that also enriched environment was giving you brain benefits, increased neurogenesis. And so there was different discussions in the field about how important is just interacting with something new, like learning how to do Sudokus or something like that, versus actually the running exercise. And so to tease this apart, they had these 4 conditions: control standard housing, the running wheel, enriched environment with the running wheel, but also without. And as soon as they took out the running wheel, you can see that there was some increase with neurogenesis, but it was just much less than you would see with the running wheel, which is, for someone like me who come from a metabolism background, does suggest that there are systemic factors possibly released by muscle in the circulation must have also benefits on the brain. All right, so to just kind of summarize, what are the cellular effects of exercise on the brain? We talked about improvement in cerebral blood flow and angiogenesis. I told you about, induction or enhancement of adult hippocampal neurogenesis. We talked about changes in hippocampal volume, which are probably a product out of more blood flow, angiogenesis. I mean, neurogenesis is not that you get a ton of neurons, that it really is going to probably change the size of your hippocampus. And another big part also is preservation of synaptic connections or formation of new synapses, especially if you think about aging, where aging basically means that you lose synapses, in the end, neurons. And then also the point, synaptic plasticity, I had mentioned that from the newborn neurons, but also, I mean, they called it synaptic plasticity. I think they also meant the, maintenance or formation of new synapses. And this all together is what leads to these improvements in cognitive function. Okay, so kind of this was an overview, the kind of what's the state of the art on exercise, cognitive benefits of exercise on the brain. So while we know a lot about what are the cellular mechanisms, what we understand much less is what's the molecular mediators. And this is really where my research program is coming in. And this is the second part of the last third of the talk, where I want to talk a little bit about the research in my lab, where we're trying to identify these underlying mediators. And we also want to find out how can we harness these molecular mediators to treat actually cognitive decline in aging in Alzheimer's disease or neurodegeneration. So what we're focusing on, as I mentioned, are these myokines. So these are small peptides, hormones made in skeletal muscle up on muscle contraction. They were released in the circulation, and they have shown to interact or do this cross-organ talk or organ-organ interaction, with fat, liver, pancreas, and now work, including our own, has also shown that they have effects on the brain. And the exercise hormone or myokine we are most interested is called irisin. Irisin is cleaved from a precursor protein, which is called FNDC5, and released into circulation. It was discovered in the lab of my postdoctoral mentor, Bruce Spiegelman. We and others have shown that exercise induces FNDC5 expression in skeletal muscle in the hippocampus, and also that, as I said, it's a systemic circulating factor, and it's increased in mice and humans with exercise. And this is a small diagram of FNDC5. So FNDC5 is a type 1 membrane protein. So you have a small cytoplasmic tail, you have the transmembrane protein, and then this ectodomain can get cleaved off, and this is the irisin. It's only 112 amino acids. It can get glycosylated and runs then around 25 kilodaltons. And one of the cool things was that irisin is actually 100% conserved between mice and humans. One question we get a lot is, how do you actually measure irisin? So since there are no good antibodies, we actually had to develop a mass spectrometry-based assay. And that was, together with Mark Jatrzewski, who is a mass spectrometrist who did his training with Steve Gigi. If people have questions about details, happy to answer. But basically, there were some questions about how do you measure it in humans, and is the human irisin the same as the mouse irisin? And to make a long story short, yes, it is. The human irisin is the same as the mouse irisin. It does circulate, and it does get increased with exercise. And it circulates at levels of like 3 to 5 nanograms, which is, typical range for like a peptide hormone. All right. So when I started working in Bruce's lab, they had just discovered irisin as a factor that was induced in skeletal muscle with exercise. It was downstream of PGC1 alpha, a transcriptional coactivator that responds, for example, to metabolic stress, to exercise. It gets, as I said, released into the circulation, and they had found all kinds of good metabolic benefits with the hormone. However, one of the first experiments I did myself was also to measure irisin in other tissues, and we saw there was a high expression not just in muscle, but also in the brain. So there was a central question during my postdoc: is there a role for FNDC5 in the brain? And the short answer is yes, there is. And we published this. This was the first paper to show that this FNDC5 pathway was involved in hippocampal effects on exercise. And it was also, I think, the first paper to really make this muscle-brain connection based on a specific hormone. Shortly thereafter, there was a publication from a Brazilian group where they showed that FNDC5 rescues synaptic plasticity and memory deficits in Alzheimer's disease mouse models. However, both of these studies had one central shortcoming, and that was the question, FNDC5 or irisin, like what is actually the active moiety? And next question, why do we actually care which one it is? Because from a translational point of view, if you're a biotech and you want to develop a drug that encapsulates at least some of the benefits of exercise, it makes a huge difference if you have a large type 1 membrane protein with different transmembrane parts and all of that, versus a small natural peptide hormone that's 100% conserved between mouse and humans. So, and to kind of answer that question, that was what I really did in my own lab at MGH. And, we were able to answer the question that irisin is a critical regulator of cognitive function. And that's what I talk now more about. So the first question is, what's irisin doing in physiology? Mainly exercise physiology. So we generated global knockout mice. They don't have irisin anywhere. They don't have it in the circulation in none of the tissues. These mice can exercise to the same extent as the wild type mice, but if you put them in these exercise, in this Morse water maze task, you can see that the wild type mice, as I already told you, I think several times over, you see this improvement in latency in the Morse water maze. You do not see that if you don't have irisin. And for the aficionados, this was the reversal of the Morse water maze, which is one of the tasks more closely related to function of newborn neurons. We also tested mice that were aged, and it was the same thing that if you don't have irisin and you age, your cognitive decline is faster. Then to kind of find out, is this really the newborn neurons? We did several things. We first checked for what's the activation pattern. We did this by looking at CFOS expression, which is a marker of neuronal activity. So we randomized, and shortly after the running, we took out the tissues. We had injected the mice with BODU to label newborn neurons. And then when you count, you can see that there was a abnormal activation in the newborn neurons in the mice that lack irisin, but no difference in the mature granular cells. I had mentioned to you that it's not just number of neurons, it's also how getting neurons integrated into the circuitry. So we tested that by using retroviral reporter virus that you can inject into the brain using stereotactic injections. And then you can really look at the morphology of the newborn neurons. And again, we saw in the wild type mice that you get an increase in dendritic complexity here measured with the Scholl analysis, but you don't see this effect if you like irisin. If you look at density of dendritic spines, where the excitatory synapses are located, you can see that there's a deficit in spine density, and the spines that you have are smaller when you don't have irisin. And even if you exercise these mice, they can't make up the difference. So, and then lastly, we actually looked at the transcriptome. For that, we actually had to develop a new approach using, again, this retroviral vectors to only sort out the newborn neurons, because even if you do single cell sequencing, you get so few newborn neurons in there. And also this technology allowed us to do this birth date labeling, where you can get one cell population of a certain age. And if you do that and we do bulk RNA sequencing on it, we see that there's a vast difference in gene expression patterns between the knockout and the wild type mice, and most of the pathways that are affected are pathways that are involved in neuronal development, and also, as you can see, Alzheimer's disease. All right, so switching gears. So this was the role of irisin in physiology. Now we're looking at Alzheimer's disease. And why are we doing that is because, this is a disease I really care about, and I showed you all the beneficial effects we saw in exercise. If we use this APP Alzheimer mouse model, there was a significant reduction in FNDC5 expression in the hippocampus. If we look at the human database, we also saw a significant decrease in Alzheimer patients for FNDC5 expression in the parahippocampal gyrus. When we cross our knockout mice with these Alzheimer mouse models and we do tests for cognitive function, this is a pattern separation task contextual fear conditioning, where the mice have to learn to distinguish between two contexts, meaning they're in these shock boxes, and they have like a different, it looks different for them, and they have to remember which is the good context and which is the bad context. And you can see that the wild type mice can do this really nicely at this age. So they have, oh, this is the bad context, they freeze more. This is the good context, they freeze less. But the knockout mice are not able to do that. All right, so now to the therapeutic application. So can we actually treat mice with irisin, and what does it do? So to do that, to really get at the question of, is this irisin, we had an adenoviral vector that only expresses the irisin protein part. All the other studies before used FNDC5 adenovirus, we used irisin AAV8. We treated the mice at an age where they already had significant pathology. So this is not about prevention of the disease. This is really therapeutics as you would do in humans. I mentioned irisin is 100% conserved between mice and humans. So if we play with mouse irisin, we're also working with human irisin. And then the last thing what we did is we injected irisin into the tail wing. So the AAV will travel through the bloodstream into the liver. The liver is going to start making irisin, and the liver is a professional secretory organ. So it will, push the irisin out into the circulation, which then leads to significant increase in circulating irisin, which is the same thing what would happen in exercise. So we did that with these mice, and then they came back after about 2 months. We did a Morris water maze, and the mice treated with irisin saw a significant improvement in cognitive function compared to the control mice. So that was really exciting. And I remember talking to my postdoc, he was really excited. And then because a PI is a PI, it's like, okay, can you repeat this in a different model? And he said, I'll do it. And then we went on and we got another Alzheimer's disease mouse model, which is more severe. And that one also had neurodegeneration. So at the time when the mice were treated, they already had neurons dying in their brain. We saw the same exciting result, that this increasing circulating plasma levels of irisin improved cognitive function in the Morris water maze. So that was, really, really exciting. So then the next question, which was actually posed during the revisions of the paper, was like, is this actually, irisin doing effects in the brain by crossing the blood-brain barrier, or is there something happening in the periphery, or how does it work? So we first confirmed that our AAV irisin does not cross the blood-brain barrier, which it shouldn't. AAV8 cannot get into the brain, and that's what we see here. A lot of FNDC5 in the liver, but no FNDC5 in the brain. And then when before we collected the tissue of the mice, we actually perfused with PBS to remove all the circulating blood from the brain. So if we now measure irisin protein in the brain, because it's not made in the brain, the only way it could get there is through the blood-brain barrier. And that's exactly what we saw, is there is irisin crossing the blood-brain barrier. Now coming back to the mechanism, I had already told you neuroinflammation is an important part. And I just wanted to remind you the two main cell types involved in that are the microglia and the astrocytes. So the microglia are the resident immune cells in the brain. And the astrocytes, they used to be called like support cells. I think now we have a much better understanding of how astrocytes and microglia are interacting, and more data on how this is really a risk factor or like an important role in the pathology of Alzheimer's disease. So that's the kind of two cell types we were focusing our investigation on. So the first thing we did is we collected hippocampal tissue from these Alzheimer mice treated with irisin. We did RNA sequencing, and then we actually did find that significant regulated pathways were these neuroinflammatory pathways. And then when we used the kind of bioinformatical deconvolution trick, we actually looked only at genes that are expressed in astrocyte or microglia and saw that, these highly activated cells, there was actually a downregulation of gene expression with irisin treatment. And then lastly, using confocal microscopy, where we actually can count the number of cells and look at the morphology, we also saw significant effects on morphology, which led us to conclude that irisin reduces neuroinflammation in APP mouse models. All right, last question. Irisin is a secreted factor. It should bind to a receptor. Which is the receptor? So previous work has established that irisin is binding to integrin receptors, and in bone and fat cells, this were like integrin alpha V beta 5 heterodimers. As you might remember, integrins are obligate heterodimers, so you always have to look for that pair. So obviously, we ask in the CNS, is that the same thing happening? And to do that, we used a super resolution microscopy. We cultured cells from adult hippocampal stem cells, differentiated them. Then we put irisin with a flag tag on it, let it bind on the cells, cross-link, wash everything away, and then we stain either for the flag tag from irisin or for different integrin receptor complexes. And every single dot you see here is one of these single molecules stained. And when we do crosstalk analysis, you can see there is significantly more irisin bound on alpha V beta 5 receptors than alpha V beta 3. We used that one because we already knew from previous study that was not the favorite receptor, or not a receptor for irisin, so therefore we can use it as a control. And then our last interesting finding was that these integrin receptors are actually on astrocytes and not on neurons. So I always had thought that there were direct effects on the neurons, but it looked like, at least in the context of here, that irisin is binding to astrocytes. There are no microglia in these cultures, so that's again something we can't tell you yet what irisin is doing with microglia. And lastly, there was a study that came out, I think, 2 weeks ago, looking at irisin in a different model of neurodegeneration, in this case, Parkinson's disease, in a mouse model. It's a so-called seeding model. What you do is you induce Parkinson-like disease in these mice by injecting alpha-synuclein preformed fibrils directly into the striatum. You might remember Parkinson's disease, alpha-synuclein is one of these misfolded proteins. And if you make these preformed fibrils, they're going to start in a cascade to form more and more of these toxic fibrils. And then what happens is that over the time, it actually, the dopaminergic neurons in the substantia nigra are dying. So these are the brown stained neurons here. However, if you treat with irisin, you can see that there was a significant protection of the number of dopaminergic neuron in the substantia nigra. And when you did behavioral tests, so as you know, the substantia nigra is important for movement. One of the hallmarks of Parkinson's disease is movement disorder. So if you do these behavioral tests for movement and coordination, muscle strength, again, irisin had rescued some of most of the detrimental effects of the Parkinson's disease model, which is quite exciting, again, to expand. That's not just something specific for Alzheimer's disease. Right, so to just recap this, irisin is the active moiety to improve cognitive function. We did this in multiple animal models. We also looked at knockout mice and wild-type mice. I didn't show that data. Peripheral irisin can improve cognitive function in transgenic mouse models of AD. It does that by reducing glial activation, and it can cross the blood-brain barrier. Okay, so that's basically, that concludes the part of the mechanistic work that we are doing. So I now have a couple of slides, which is supposed to kind of get us into the discussion, based on some of the questions that were submitted before, and then we can also open up to the audience. All right, so how much exercise do I need to do for my brain? So I just wanted to kind of let first speak the official authorities, so the CDC and the American College for Sports Medicine. The current recommendation is a minimum of 150 minutes of moderate exercise or moderate intensity physical activity a week. If you do more intense exercise, you can exercise less. However, to get substantial health benefits, and remember in the dementia, 4,000 steps, 25% reduction, 10,000 steps, 50% reduction, you need to do more than 300 minutes. And, of course, muscle strengthening activities. I would like to add my take on that a little bit. More is better, but every step counts. So that's one of these things sometimes people, ask that maybe don't have access to a fancy gym around the corner, like, does it make sense to even just use a step counter on my phone? And I can tell you, yes, the more steps you can do, or whatever it is, what you can do more in your daily life to be more active will have an effect, especially when we talk about prevention in an aging population. Another important point is stick to what you can stick with. I always get asked, oh, so which exercise should I be doing? Is it hiking, biking, swimming? I don't really like swimming. Do I have to swim? You should do exercise modalities that you actually enjoy so that you actually can do them. That's what we call then the compliance. So, as I said, I really like hiking, so I do a lot of that. Other things, I'm not so much big fan of, I do less from. And then again, the other thing is also do what you can do. I would love also, or many of us probably would love to go to maybe a nice, sophisticated yoga studios for several hours a week and really practice there. But if the best you can do is a YouTube video with yoga exercises for 20 minutes at night before you can go to bed, that's much better than not doing any exercise at all. So also be realistic about what you can do and rather accomplish what you want to do. So that's kind of like my personalized take-home message on that. All right, how do I know my brain is healthy enough? All right, good news and bad news. Bad news first. So we don't have like a blood test or something similar we have for heart disease or like for lipid profiles where I can either measure your blood pressure or take your cholesterol level and tell you hey, your heart or your blood pressure is that much compromised, or cancer screening, where you can look at that. So we don't have that for the brain yet, and that's something we're working on at the McCance Center. However, what we do have, and that's really one of the accomplishments over these last 2 years, from the leadership from Zaina Chemali and also Jonathan Wrann, is using all the data that is out there, that all these lifestyle modifications actually can help you at least prevent the development of a lot of brain disease, dementia and other things. So, and they combined and came up with this scorecard where you can look at different categories, different physical measures that they actually can take, lifestyle factors, even social emotional connections, because we know stress is one of the things that can be detrimental to your brain health. And then you can come up with a score, and it even gives you some suggestions what you can do if you found deficient on certain aspects, how to improve that. And it's freely available, so anybody can, if you go on that website, you can actually take a check for yourself. All right, and another thing. When can I get the exercise pill and finally stop worrying about doing exercise all the time? So obviously I had mentioned that we want to, turn these benefits of exercise into a drug treatment. However, exercise is so complex and has so many beneficial effects. I mainly talked about the cognitive benefits today. However, there are also important effects on, on mood, reducing, stress reduction, improving, anxiety and depression. These other aspects. I had mentioned all the other effects on muscle strength, on cardiovascular health, on metabolism. So the idea that you're going to come up with one pill that encompasses everything great, all the great things that exercise can do for us, I don't think that's a realistic expectation. However, there are many patients that are, they're limited in their capacity to exercise to a level that they would need to do to really get at these brain benefits. I have mentioned, you have to really push your aerobic exercise to get there. So I think for these patient populations, there is a critical unmet need of developing new pharmacological interventions. And exercise is already doing what we really want any intervention to do for the brain. So I think this is a really good starting point. And lastly, we might even want to push in the treatment in an Alzheimer patient where already we lost a lot of synaptic connections, where there's neuronal death, we might want to push beyond what exercise can do. So I think that's kind of my take on that question. So, all right, summary. I have told you about that exercise can improve cognitive function and prevent dementia. We have discussed different important cellular mechanisms of neuroprotective effects of exercise. And I introduced to you the exercise hormone irisin, which we identified as a crucial molecular mediator of the cognitive benefits of exercise and see as a potential drug target for Alzheimer's disease in the future. And lastly, I would, of course, acknowledge the people in the lab who did this great work. And it was really a team effort. It was spearheaded by 2 postdocs, Rashid Islam, who is now working at Flagship Pioneering, and Sophia Velaris. They were supported by a great team of research technicians, graduate students, and also rotating students, undergraduate students. So, you know, fantastic team to work with. I'm really happy. I'm very proud of them. We had, of course, also great collaborators. You know, this is never the work of just one lab. You might recognize some of the names there. And, you know, and of course, I thank my funding sources. I mean, you had already mentioned the Spark funding from the McCance Center, which was part of that, because without the money, you know, we couldn't do the work we need to do. So with that, I'll end, and I'll be happy to take any more questions. Do you want me to close the presentation to change that or leave it there? Okay, good. So the solo Zoom things is kind of nice if you go on. Yes, and I heard if you stand under these squares over there with the green light, he brings the microphone. Oh, sorry.
Nice talk. Thank you very much. So I have a question regarding aerobic versus weightlifting exercises. So you mentioned that the aerobic exercises are typically more beneficial, but I was surprised to see that irisin is produced primarily by muscles, and so the weightlifting exercises aren't that useful. So can you explain a little bit on that?
Yeah, so the muscle gets exercised in 2 different ways. If you do aerobic exercise, you know, the running, you also put strain on the musculoskeletal system because, you know, you create need for nutrients, you know, and all of that. So there are, how do you say, adaptive responses in the skeletal muscle also with aerobic exercise. They are different from the adaptations you see in muscle strengthening exercise. And a very good example on a molecular level is PGC1 alpha, which comes in 2 isoforms. So it gets induced by exercise in both modalities. And one modality gives you alpha 1, and that gives you then irisin. And then the other one is the alpha 4, which is then in the resistance exercise. So this is how basically the muscle is on the crossroad of both of them. And some of them have more cognitive benefits than others.
Thank you. I have one more question.
Yes, go for it.
How you mentioned that the beneficial effects of irisin could be through the anti-inflammatory effects or by reducing neuroinflammation. So have you tested for the anti-inflammatory effects in general or even in the periphery of irisin?
This is work we are doing right now in the lab to kind of understand how much of that is a specific, you know, Alzheimer disease immune response or modulation of the neuroinflammatory response in Alzheimer's disease versus a general program. You know, Joanna is working on that in the lab, and I think we are seeing effects that are kind of saying it might be generalizable across just Alzheimer's disease.
We have a hand up. Mohammed, would you like to go ahead and ask your question?
Is your mic on? I'm not sure. Is it on?
Hi, yes. Mohammed, would you like to ask your question on Zoom?
Yes, it was a great talk, and I have a couple questions. The first one is about the astrocytes. So when you see that the irisin has the receptors on the astrocytes, after treating like the astrocytes in vitro or even if in the hippocampus, what could be happen in the metabolomics of the astrocytes? Because they like the more like supporting the neurons like by the like glucose homeostasis or energy expenditure. And what do you think that irisin can impact on their like the metabolomics? Like if we analyze their metabolomics, what could be happen? This is my first question. And the second question is about the Parkinson slides. And I read that paper last week, and the question that I have, like the substantia nigra has the circuit into the striatum that can control like the movement, and also it's impacted in the Huntington diseases. So in your bulk sequencing slides, the pathways that you sort out the Huntington diseases was standing there as a second or first, like the correlation with the FNDC5 and the bulk sequencing. Do you think that FNDC5 also impacts like the as a medication, or I don't know, remedy for the Huntington diseases? Or thank you.
Okay, all right. I'll try to unpack. There were a lot of things in this. Try to unpack. So going back to the astrocytes, so there is a lot of work from very established labs in the field to show what are astrocytes, how they are actually playing a role in the pathology in Alzheimer's disease. So one of the things they are involved is amyloid, metabolism, like the making and breaking down from APP precursor proteins and other products that are then contributing to plaque formation. There is work gonna be coming out hopefully soon that really addresses specifically the role of irisin and astrocytes in that. So since it's unpublished, unfortunately, and it's not my own work, I'm not free to speak about this in detail, but this is at least one way how astrocytes and irisin could have a work in Alzheimer's disease. The other what I had mentioned is we also know that astrocytes talk to microglia, and microglia are very important as well, and like in mediating neuronal death. So it might also be that there's a crosstalk between astrocytes and microglia that is detrimental or getting corrected with irisin in that model. I think that, and then for the metabolism, if you think about, and we haven't delved deep into how metabolism in neurons is regulated, but if you look at the gene sets that making up the pathways that were regulated, especially Parkinson's disease, a lot of mitochondrial genes are in there, because it's known that mitochondrial dysfunction is a very important factor in Parkinson's disease. So it might be that another effect is also regulating metabolism. Now, it was a pretty big paper, and we explored mainly one mechanism of action. There might be more than one. And then switching gears, towards the Parkinson's paper, I think I already answered part of the crosstalk there. Huntington's disease, that's an interesting question. The PGC1 alpha also plays a role there, but if I remember correctly, actually, there is a problem with hyperactivity also in Huntington's disease, and over-exercising might actually be detrimental in a Huntington's disease mouse model. So we haven't tested it, irisin and Huntington's disease model, so I can't tell you if it's gonna, which way it's gonna go, but there is definitely a connection between PGC1 alpha, Huntington's disease, and exercise that's worth exploring more.
Thank you.
There's more question over there.
Thank you. Beautiful talk. Really enjoyed hearing about all of your work. I had followed the irisin story when it first came out, and then hadn't seen as much recently. So it's just wonderful to see how it's developed and how you've brought it to the brain. I'm curious to hear if we know anything about dosage of irisin and how that may affect response to Alzheimer's disease in models, whether there can be toxicity if you have superphysiologic levels, things like that.
Yeah, thank you very much for that question. It was a big paper. It took quite some effort and time to put it all together. So back to the dosage question. So the levels we actually reached with the AAV application are pharmacologically, they're not physiologically. And one part of that is, you need to have enough to also cross over to the blood-brain barrier. This is something that we are now really want to drill down on, and the funding from the ADDF will help with that, to really go a little bit more into like pharmacological research to understand, developing markers of, target engagement to kind of say this dose of irisin is going to give you as much effect on that, because that's a crucial part in the drug development. What we do know is we have treated mice for up to 2 years as we injected the mice with the irisin AAV I forgot how old they were, probably, at 2 to 3 months old. We kept them until almost 2 years old. The data didn't make it into the paper because we had a very small N. We saw improvements in cognitive function with irisin in those aging mice. I think was an N of like 5 or 4 at the end. But we didn't see any detrimental effects in the sense of, we looked at body weight, spontaneous locomotor activity, sorry, in these mice, so which are like pretty, like readouts where you can tell if the mice overall is doing that. So like 2 years of irisin application didn't do anything. We did like some smaller scale pharmacological studies where we looked at, did a clinical chemistry panel, like what they do usually also for toxicity testing, where we looked at enzymes for kidney function, liver function, calcium homeostasis, other things. We didn't see any significant effects there. So again, no effects on body weight is there. Of course, we have to, someone has to do like the the standard in biotech for toxicity studies, to be, absolutely fair. But so far we haven't seen any weird toxicity. And on the other hand, in the knockout mice where you don't have irisin, when you just have them like the knockout mice sitting in a group house cage and they don't do anything, we didn't see any huge effects there. Again, we only saw effects when we stressed the mice, we exercised the mice, we aged them, or in the Alzheimer mouse models, which my understanding is also that irisin is probably a plasticity factor that comes into play when you challenge the system. Again, that kind of, I think, goes hand in hand. And then again, the neurological effects were the strongest. Someone had also asked about metabolism. Again, the effects in the brain were the strongest in these experiments.
We have a question on Zoom from Javiera. Thank you for the great talk. Did you look to see if irisin also improved mood or depression or anxiety-like behaviors in the mice you were looking at?
Good question. We have not done this yet. I would almost be surprised if that's not the case. I had told you before that PTC1 alpha is the upstream regulator of irisin, and there's really nice work from George Ruiz' lab. He also trained with Bruce, where they showed that PTC1 alpha is one of the muscle factors governing mood and stress. So, and since irisin is downstream there, I would assume that is. We did assays for anxiety at baseline, and we didn't see a difference. But for most of these models, you actually do stress the mice, and so I think I would bet something on that there will be differences.
Thank you. Do we have more questions?
Yeah, 2 more.
Thank you, wonderful talk. I would like to ask if you have tried or considered to administer irisin directly to the brain using intracerebral vascular injection. I mean, in the form of protein, isolated protein. Thank you.
So I didn't show the data. So what we did is we delivered it, the AAV8 directly, serotaxically. And in that case, we targeted the dentate gyrus. And there we saw then effects on pattern separation behavior that was dependent on newborn neurons. We have not done, larger scale trials with the protein. I think that would be an interesting question to kind of compare the effect size you get from central application versus the effect size of peripheral application. So the one thing is what we do like for the drug development is the hope that peripheral application is sufficient, to cross the blood-brain barrier, which is, as you probably know, a very important or could be a significant barrier in drug development. If you have a compound that, if given ICV, works great, but if you have to give it in such concentrations peripherally to get central concentrations, that you get toxic effects in the periphery from that, which is, by the way, one of the reasons why some of other factors, a lot of the growth factors, unfortunately didn't make it to the clinic. IGF is one of them, has great effects also on neurogenesis and vascular, but it also is pro-growth and can cause cancer. So I hope that answers the question.
So are you looking at using irisin in any way as like a progressive biomarker, like response to irisin, as well as potentially like a more clear diagnosis for Parkinson's disease?
Really good question. I don't know if you remember the slide that I had shown about the mass spec assay that we developed. One problem we are still having is there is no good commercial antibody to measure irisin levels very easily. We tested all the ELISAs that were out there in our knockout mice, and we didn't see a difference between wild type and knockout mice. So irisin seems to be measuring, the irisin antibody seems to measure irisin, but probably also something else. Also, the assays were usually an order of magnitude higher than what we measured with the mass spec. So there's something going on. So it would be great to find out how good irisin as a staging or diagnostic factor is, but we would have to get like an antibody tool to have it in an ELISA, because doing it with mass spectrometry is just not feasible. It's very expensive. It's very labor intensive. That being said, there is work on Alzheimer's disease from the Brazilian group, where they used some of these commercially available ELISAs, and they were trying to make the case that they did see a correlation between irisin levels and amyloid pathology and cognitive function. Given what we know about the ELISAs, I always would just caution people, to take this with a grain of salt. Is that? Yeah.
Patients, has this been used on non-mouse models?
If we have used irisin in patients, no, not yet. I mean, as you, there's like a how do you say? There's a process you go through before you can test something that is working in the mouse. You can do that in the humans. So that's one of the, kind of the next steps that we want to do, also in our lab, is to really get irisin from an experimental drug in mice to like an IND kind of version that you could test in humans. So, and that's something I also want to caution. I do sometimes get really heartbreaking emails or letters from family members where their father or their mother is suffering from dementia and asking us, can you please send us some irisin? And it's really heartbreaking, because it's just not something we would be ethical to do at this point. So I just wanted to put this out there.
Sorry to just I'm just wondering if in mice it's able to be like discerned when they do these tasks, if they have better internal cueing, because you know how that's like a difficulty for then even looking at like how this would integrate in like to the brain system for like autonomy and not drug reliance, as in once you introduce irisin, are then they able to internally cue, and then obviously then it gets integrated with the motor planning and motor learning, and then instead of having that memory and almost Alzheimer's like symptoms and one cardinal or one symptom of Parkinson's that can lead to a diagnosis, as in like is there some sort of autonomy that gets integrated with irisin?
That's a really good question. We haven't really mapped on a on a circuit level, which, in the Parkinson's model, which are all the circuits that are, affected. We do know that, that there's a preservation of dopaminergic neurons in the substantia nigra. I think we also looked at some parts in the striatum, how that is, but how that then again plays into the cognitive function. I think that's what you were like, or cognitive benefits. We haven't crossed that bridge yet. So I think that sounds like a really interesting project actually to do that, to kind of again understand how this is related. And what you just mentioned, maybe for the general audience, it is very common that you have what is then called Parkinson's disease related dementias, that patients that are getting diagnosed with Parkinson's with the motor symptoms first, that they later on go on to develop cognitive problems as well. That's in the dementia part. And on the other hand, there is a Lewy body dementia, which usually is more getting diagnosed as cognitive function deficits, and the patients go on to develop Parkinson-like motor symptoms later on, and that's kind of, I think, where you kind of at this cross section where you were asking about. So I think it would be very interesting to understand how irisin is working at that cross section, and if maybe one is helping with the other or not. So I think it's a great suggestion for future project.
We have one more question on Zoom from Eduardo. He asks, I was wondering about how irisin interacts with the BDNF responses induced by exercise.
Yeah, good question. So in our 2013 paper, we really just focused on BDNF because at that time, this was the leading neurotrophic factor that had been connected to almost any of the exercise benefits. It was like, okay, it's BDNF, it's BDNF. So that's why we specifically looked at BDNF. In our 2021 paper, we really wanted to take more an unbiased approach. What is irisin doing in the context of Alzheimer's disease? And in that context, we found that the anti-neuroinflammatory responses were much stronger than the effects that we saw on BDNF. Not saying that one is more important than the other, or one is not having to do with the other, but that was just what we saw in the second study, which I also find encouraging that it's not, I mean, it would be unlikely that, you know, you have these like very intricate systems where one hormone is talking to other things, that it would be just like only one downstream target, and this is the all there and there to be.
Thank you. So with that, I think we will close off the event. Thank you so much for the wonderful talk, and thank you to the audience for your wonderful questions.
Thank you very much.